Good. I think we can start. Welcome, everybody. I met yesterday evening for the first time Jonathan Barikiewicz, and I have to say that he's not only a very, very nice person, but also a very good seller, because he told me, I found very old trees in Chile. And I said, oh yeah, how old? And he said, I will tell you tomorrow. So that's why I'm here. Otherwise, I wouldn't be. Patrick will share this promising session. So Pat, the floor is yours.
Yeah. Thank you very much, Paolo. As I said, I don't know Jonathan very much, but I would say that in the 3 days I had time to interact and speak with him, I would say it's a very interesting guy. He's interesting to me because he's working really at the intersection of two different worlds. From one part, we have the cells, the NSC, mechanism, and then the other part there is the climate system model or Earth system model. So he's trying to working and combining all these worlds together. And I would say that in addition to that, he has very interesting keyword that I attribute now to him based on the discussion we have, which are xylogenesis, flux tower, tree ring. Is also somehow passionate and interested a lot in the underground, so roots, soil interaction, isotopes, drought, and of course the Earth system model. So it's a very diversified, broad scale of different interesting topics. And I think in his presentation, he will show us a little bit how he combine all this. And I hope you will find that also interesting. And yeah, I'm looking forward to hear what is going to tell us. So the presentation is actually titled Tree Ring Constraints for Global Land Surface Models.
Thank you very much, Patrick, for the invitation, and Paolo. I work at the intersection of dendrochronology or field of dendrochronology and climate modeling and ecosystem modeling, which in the end is air system modeling. And so I will be talking to you, showing you more or less the work that we have been doing and the difficulties that we have, and also the opportunities, and a bit I will show you some results we already published and more or less where we are going. Dynamic global vegetation models or land surface models are within the Earth system are the models that represent the land surface. So all what is green, the land, the vegetation, the desert. And then there are Earth system models have also a component, ocean component, an atmospheric component, ice component. So these models are the one that model the land, is a subcomponent. And this model underpin all the climate projections of the IPCC. So every air system model has a land surface model inside. And then, so the role of land surface models and dynamic global vegetation models can be used to monitor long-term trends in carbon, water, or energy, and in vegetations in the land surface. Also to attribute the causes of trends and variability in these fluxes, and also to predict the changes into the future under the different climate scenarios and within these air system models. So this is how one grid box is represented with different land covers, and then we simulate the different fluxes of these land covers and the interaction with climate. And so when we simulate from the start of the Industrial Revolution until the end of the 21st century, when we simulate the biomass, we see that different land surface model, they really diverge a lot toward the future. So there is a big uncertainty in the projection of these models. And then the data behind this model have been developed and parameterized with different kind of observations. So on the right here, we have the number of time series that are available, and on the left, the temporal depth. So manipulation experiments like phase or Klimani and etc., warming experiments are very important for the parameterization of the processes that are in these models. But they are short term and a few years, and there are just less than 200, let's say, overall. Then we have FluxNet side that has revolutionized a bit the modeling simulation of the fluxes, and there are about a couple hundred of stations around the world, but they go just a few decades now to 2 decades or 3 decades, the longest record. Most of them are younger than 10 years. And then atmospheric CO2 records, but again, they started in the late 50s, and there are a couple of hundreds over the Earth. And then the major stream here is Earth observation data, where we have many for every pixel high resolution. But again, since 1980 onwards, and then forest inventories, we have also a lot, but again, they just are for a few decades. And so then the motivation of my research here is that these models show this big uncertainty in the long-term response of the land carbon cycle, and that feedbacks into the CO2 concentrations, and then in the speed and magnitude of climate change. So here we see that, for instance, these models that have been evaluated and developed with these short data streams diverge wildly to the future. So some models show that the global biosphere is going to be a source of carbon to the atmosphere, and others that it's going to be a sink. So it's a wide range of answers. And then the origin of these uncertainties can be attributed to model structure, parameters, and then the drivers that we use, the climate drivers, at least for the historical period. But then inside the model structure, I argue that one main control, at least for the component of tree growth, is that there is a flawed growth assumption. So in these models, biomass growth is a direct dependency on photosynthesis. So there is no wood formation. And that has many implications, because the biomass growth is basically equal to the gross primary productivity, to the photosynthesis. And this is the well-known source-driven growth paradigm that Christian Kerner and Fatici and many others have criticized in these models. And that there is now, it seems, some other groups that are also going in the way to replace this paradigm, to integrate it with the sink growth paradigm. And I will talk a bit more about that, but this, in terms of processes, this is one of the main, I would say, complication of the models now. And then the other is that there is a lack of long-term constraints. So we saw that most of constraints, the longest go 3-4 decades, 5 decades, more or less, few. And then that is also a limitation. So, and that not having these long-term constraints also make our parameters more uncertain. So then we argue that tree ring hold the key to solve these two things at the same time. I mean, to improve the process, to replace the source-driven growth paradigm, and also to have the long time series that we need to improve the parameterization and the responses of this model at long time scale. So if we add tree ring, you well know that we have a couple of thousand, and then we go back in time a long time. And then if we see it in perspective, now we see here in green from 1500 to 2000 on top, the basal era growth of Fitzroya tree in forest in southern Chile, for instance, in green. the blue here is the CO2 concentrations, and then we see how growth has increased a bit hand in hand with the in this case with the CO2. But then we see like phase, eddy covariance, and satellites observation, their temporal depth. So basically, these data give a view when the system was already when CO2 was already high. We don't capture a gradient in responses of CO2. So these tree rings can help us to constrain the biomass production in the model, and also transpiration at long time scale. So they give simultaneously they solve these two problems that I discussed before. But there are challenges with that. So we have been working in this, and there is in general a negative view of tree rings in the global change community. So papers or proposals, you get quite, the reviewers are quite skeptical when you go to the global change community. For instance, here is a comment of a panel, and they say, we acknowledge the general difficulty of disentangling multiple climatic and biological influence on tree growth data that challenge their use within this type of research with the integration with models. Because for models, you really need to work with processes. And in the tree ring community, we have been, I would say, very empirical. We rely a lot on correlations, and then we have trends. Trends are quite complicated to deal because of the different biases and spatial coverage biases as well. So there is a skepticism, and it's a tide that is difficult to turn. But here I argue that that tree rings really are the key thing that we need to improve this model, at least to reduce a good chunk of that uncertainty in the future. So if we think like in three factors, A, B, and C, A, it would be the global change factor here over time. So we see nitrogen deposition increase in CO2, drought severity and frequency, BPD, temperature, and then we see in tree rings, we go straight to C, we see that there are changes. So for instance, wood density, at least in Europe, is decreasing. Tree ring width doesn't change much in many places overall over time. And then we see that growth synchrony has increased in recent decades quite significantly. And then we see also that water use efficiency has increased strongly, but in recent years, that rate of increase is slowing down. But then how do we interpret what we see here and with what we know, how the global change factor had changed, and are all these processes? So here I go back to Harold Fritz, and then he has this image that most of you probably have seen at some point, and then it's how climate and then physiological processes filter climate and result in a narrow or wider ring, passing by how the sink work, like the cell and the cambium work. And I call that physiological dendrochronology and in monitoring sites. So because we should be able to monitor and integrate all these processes in the same site, and that will help us to know more about the processes and go a bit addressing the question that the reviewers comment I showed you before. So I argue that is here where we really need to do work in order to put this in the models, and then we will be able to go from global change factor to the responses and disentangle the responses. And to go about this, to integrate tree rings and land surface models, I see two paths. The first path is that we take a current land surface model where and we use available tree ring data like oxygen or carbon or ring width, and then we compare them but this risk somehow that we get a right answer for the wrong reason, because these models, they are based, their growth, biomass growth is based just on carbon source, so photosynthesis, and then some have a pool of labile carbon. And so the work that we have been doing in this path is in the first paper, paper that we just published in Biogeoscience, and then another paper that is accepted and will be published like in a few days in GMD as well, open access journal, geoscientific model development. And then there is a second path where we use an improved model plus the true ring data that we already have globally, plus phylogenesis. Then we would have the right answer for the right reason. And so it's adding phylogenesis here. So then we will have a growth as a function of the carbon source and then also the zinc. And in this line a bit, the work that I am doing, I wrote an ERC proposal, which is in evaluation now, to do just this part. And then I also have a monitoring site in southern Chile, which is called Alerce Costero. But so here there is another interesting point here, what I argue. So when I was talking about the processes before, so to go from 1 to 2, we need physiological dendrochronology, which is what I call the monitoring and understanding of all these processes, how physiology is linked with growth in ringweed, how we can link the source and the zinc. My approach somehow is moving from this side toward this side by adding the zinc, but again, developing the process understanding. Then if we think in terms of processes, we have an observed ringweed is the aggregation model of Ed Cook, where growth is as a function of source and zinc. So the climatic signal, part of the ringweed will be explained by the climatic signal, size-related trend, endogenous disturbance, exogenous disturbance, and measurement and some unexplained process, like memory, for instance. Growth memory is an unexplained process here. And then we have to have a land surface model that has at least some components of this model. So, and then Orchidea has a climate-driven growth, has a size-dependent growth from allometry, so the equivalent component. It has within stand competition as well, and also has the natural and anthropogenic disturbances, which is nitrogen deposition and increasing CO2. And then there are model errors and unaccounted process. Currently, an unaccounted process here is the zinc, the major thing. But this is how the model it is now. And what is Orchidea? Orchidea, I said before, is the land surface component of the IPSL air system model. You have models of small scale population dynamic. For instance, ED is one of these models, individual based model, where you simulate each tree in a forest stand. And then you have large scale global biochemical models where you represent vegetation just as a layer of green stuff, green leaf, and some biomass, and that's it. You don't see individual trees, and that is Orchidea, the traditional, let's say, called trunk, and then Joule, CLM, and many other models. But then Orchidea tend to go towards an individual-based model because it drop this simplification and add size classes. So then we will have circumference growth for each of these size classes. And so then in the model, Orchidea, the photosynthesis is the Farquhar model with the Bolberry stomatal conductance. Then ringweed is simulated for each circumference class. Size class is just the difference between the circumference between this year and the previous year. And so it's radial growing increment. And carbon isotope, the classical model. And then oxygen as well, Turing cellulose oxygen, and as a function of leaf water and source water. which are simulated in the model. And so yes, some first result of this, the approaches that we are going. I was saying this is the first path 1, where we take the model as it is, and then we use the data that there are, and we try to improve the model with those data. So the ITRDV is the gold mine, but it has these complications that has precluded or generated this negative view of terrains. But we think that even with that, you can still derive benchmarks for diameter growth of trees. So the first one is the component of site-dependent growth. So you have the age here of a tree in the top, in the x-axis, and then the y-axis is the ring width as a function of age, and then the blue is the model, and the black is as observation, for instance. And then the diameter increment of mature trees as well, you can derive curve of diameter increment with age. And then you can also have the diameter increment just of the young part of the trees. And then you can also derive other benchmark for extremes. So you can see how does the model simulate more or less well the growth extremes. And so the ITRDV is still useful. Second, we try to combine the growth and physiology, because as I said before, in order to understand growth, we need to link it with physiology. And in that direction towards this physiological dendrochronology, which I call, I work around, I thought hard about how we can link growth with the physiology that the isotopes comes and the growth that the ring width tell us. And then I created this triplet representation, which facilitate the linking growth in a given year with the physiological responses that isotopes tell the trees have at the leaf level. So this triplet can be also derived for the observations. And then if you simulate the three things, the ring width and the isotope in a model, you can compare the model with the reality. And here we compare Orchidea and made in ISO. And here we see that made in ISO has a very different response surface. And even when it has been calibrated very well for ring width, here is the gray line is made in ISO in each of the panels. And for individual parameters, it performed very well, but then when you combine it, the performance is very different. So in this case, Orchidea with default parameters, still as the two are source models, the Orchidea tend to be a bit closer to the observations in the relationship between leaf physiology and what goes to radial growth, at least in this site, oak site, temperate site in Fontainebleau in France. And then the third line that I was saying, it was having monitoring. And then I have a monitoring site here in southern Chile. This is the temperate rainforest of South America is right here. And this is a projection of precipitation and for the end of the century. And we see here that there is a drying hotspot, and it's one of the strongest drying hotspot over land, and where the models with high model agreement. So we have the Mediterranean as well, but not that strong as it would be there. So we are in a drying hotspot right there. And so it's a drying biome already, and the climate space for this species is shrinking for this rainforest species. And then, for instance, here we have from 1900 to 2000, to the end of the 21st century, we have in black the PDSI, so summer drought here, and then we have a 10-year drought that is here. We call it mega drought. And then we have the range of PDSI from two models. And then we see that the observations tend to follow what the projection is a strong drying. So we are seeing some early warnings, some tree dieback, dying trees, and peat bogs are drying. And in this site, to understand this, we are monitoring eddy covariance. We have the core and eddy covariance, and then we have soil respiration, stem respiration, subflow, litter fall, tree rings, dendrometer, forest inventory, and stream discharge. So we can really work out well the water and carbon cycles and how it links with tree growth. And so this is set up of the site. Here is forest, and then we have the tower here. In the footprint, we monitor tree growth. So here is a view of the tower, and my brother there downloading the data. And here it's my mom collecting samples, microcores in the trees in the flag tower footprint. So we have got already some view of very preliminary view of the phylogenesis there, and then also she measures the shoot growth of our conifer, which is Phythiroia cupressoides. And so with that, we have been able to derive the seasonality of fluxes. Here is the net carbon exchange. So blue is that the ecosystem is capturing carbon. So as wider the blue, as higher is the carbon capture. And when it's white, it means that it's a source or is near neutral. So here we have the annual cycle in the southern hemisphere. So the summer is December, January, February, but we see that this ecosystem is active in the spring. Then we can link it also with the wood formation, with our work that we have done with the sampling of phylogenesis. We find that the peak growth cell division is in summer solstice, 21st of December, and then of course the carbon deposition occurred 1 month later towards the end of the summer. So the phenology for growth and the ecosystem are different. And with this, we can really understand the process, how the growth process links with what we see at the ecosystem level. And this is a collaboration with Sigrid Radgeber and Patrick and Marina Fonti. And here are some samples that I brought now. And for Phythiroia cupressoides, which is the species with the oldest trees in the world. And then Nosophagus betuloides is another species, which is the southernmost tree species in the world. So 2 extreme species. Then here for Paolo, start to wake up, and here is coming the stuff, the good stuff. So there we have this big tree. It's called alerce milenario in Spanish, millenary alerce, and this tree was discovered in around 1972 by my grandfather. He was a park ranger, and I would say that this is an almost immortal witness of global changes. With all this that I show you, I just want to say that Turing data have biases, but at least we have shown that there are workarounds, and then that long-term benchmark for biomass growth, transpiration, and water use can be derived from this data. And then the other point is that until now, very few models can explicitly simulate Turing parameters. So Orchidea, CLM, LPX Bern are other models that simulate isotopes. And Orchidea is the only one that simulate at least the ring width and isotope together. And then, so an important point here is that in order to use Turings, we need to change the models. We need to change the land surface model. So we need to replace the source-driven growth paradigm. And then in order to change the models, we need ecophysiological dendrochronology, which is how we link the processes, ecophysiological processes at the ecosystem level or the leaf level with the sink activity.
Thank you very much, Jonathan. That was a very comprehensive and very interesting presentation. I think people will join me in saying that I was right. You're really a very peculiar guy, and just the fact that you were able to involve your mother, your brother, your grandfather in what you are doing in Chile, I think it's a sign that you are peculiar. So I find that also a specificity of what you are doing. Please.
I have just a very short comment. Don't publish your alerce milenario in some other journals, but in Dendrochronologia.
Okay. Yeah, thank you, Jonathan, for your talk. It was very comprehensive, like Patrick said, especially for non-modelers. So we really could follow all your slides. I have a question or a comment about your monitoring side with the ecovariance flux tower. You show us the net carbon uptake, and then you also show us that you're monitoring the phylogenesis. My question is, I don't know, do you consider to convert this phylogenesis or these cell wall thickening processes to actual biomass, like having wood anatomy and convert, trying to convert the dynamics of the stem with grams of carbon, like Enrique Cuny did in his paper, I see here as well. Or another question would be if you are considering you are monitoring non-structural carbohydrates also to assess, because we are assuming that what is from what you measure from the tower goes directly to the stem, but there are other parts of the carbon that might go to other places. I don't know, what is your following ideas on that? I think it's very interesting.
Yeah, so that's a very good point, and thanks that you raise it. And I think that is one of the main objectives that why I thought in doing this monitoring. This monitoring is already from 2018, so we have 3-4 years, and it's exactly for that, because we can follow better carbon from the leaf or the ecosystem level going to the tree level. And then I didn't talk about the water use efficiency as well, because you can get at the ecosystem level and you can get also from tree rings. So and the striking thing is that this process occur at different periods of the season, and that tells you that the drivers or the environmental responses are different. So definitely what we want to see in the end, the currency for models is carbon. So it is refining the sink by doing what you mentioned, that what Cyril and Patrick have been doing is convert this phylogenesis to carbon, actually, which that would be the thing in the end that for the model would be interesting.
But do you also have data from NSC or non-structural carbon?
NSC, we measure it, but campaigns only. So we have still, we are at this moment, we are just able to build the phenology, but not really over time. So we can build a bit seasonal cycle of carbohydrate storage and growth and also net uptake. But we are going there. And that's why I think it's where all things converge in a way, all the approaches.
Very nice, thank you.
And maybe I say something wrong, Jonathan, you'll correct me, but I think what is also interesting is that Jonathan is also looking a little bit at the root growth. So it's not only focusing on the stem, which is another important component that is usually forgotten. So I think also this is something is quite strong in his approach.
Yeah, I didn't mention it. Yeah. Thanks for bringing it, Patrick. I didn't mention it because I needed to simplify. But so when you touch the simulation or the sink of carbon in the tree, let's say we do it to the stem and we put the model of sink growth or cambial activity, we will affect the allocation. So in order, you really need to have a bit of a constraint, and I am looking at ways how with the same tree ring I can still constrain a bit the allocation, and also because the global question is where does this extra carbon with increasing CO2 goes. So that's why I'm also, I would say, strong interest to look below ground and integrate with above ground.
Okay, I saw for a while a hand from Cyril. I don't know if the answer was already given and then you deleted it. Or if
yes, because you asked already my question, which was about the roots, because this was to follow with Elizabeth's question, and yes, non-structural carbon is really important, of course. Yeah. But also most of the time we also forgot the roots, and we are in our flux tower site in northeast of France. We are at this point also that we need now to probably to monitor what's happening in the roots, And for me, it's not easy because it could be roots. They could have a strong phenology, or they could grow all the year long, depending of the site and the species. And so it's quite difficult to say in advance. And your site, Jonathan, is really peculiar, really interesting from a xylogenesis point of view, and also, of course, for the flux tower. So yeah, I was wondering if you, for example, how many years do you have of root monitoring and which kind of data you have?
So now I did a chronology for roots and stems. So that's work in progress, and that at some point I will sort of build a picture with that. But and then I started to monitoring roots with dendrometers and stem the previous season, and already like piloting, and actually it is really interesting. So it really showed things that you don't expect, and when you go to dial cycles, and you can really understand better the tree, let's say. And so now we are applying for a grant. We hope we get it, which will have a mini rhizotron for long-term monitoring in the site too. So because we need to connect coarse roots with fine roots. And so this is why I have contacted you guys, because here we need to advance this integration, and we need several sites. And if we could actually eventually build up a monitoring with common protocol, it would be very nice, because we would produce we will really get new stuff from there. I think there is a lot to learn.
Okay, thank you. So we keep in contact on this.
Yeah, yeah.
Yeah, coming back to the alerce millenario, what kind of conditions, ecological conditions, were characterizing this site? Because earlier today, discussing with John, we thought about Bristol pine, very dry and very arid. But if I remember the alerce I saw, it was completely wet and pluvial, so rain forest.
Yeah, it's 5,000 millimeters of rain, between 3,000 and 5,000, and it is growing at an elevation of 700 meters above sea level. So temperature is mean temperature around 10 degrees. And then what is particular of this guy is that normally this species grows in the summit, where soils are very thin and there is no competition from other broadleaf species. And this guy lives with the broadleaf species. It's lower than the main forest. So it's having a deep soil, and it has better, in a way, and it's protected. It's the only tree in a kilometer radius. It's the only alerce tree. So it's really a guy that survived there and has been for 5,000 years now, living there, and no fire, no disturbance. Many other forests have been burned there, but this one escaped because it's in a protected spot. So it's a unique from every sense.
Yeah, thank you.
Thank you, Jonathan, for the nice presentation. Thank you for everybody for the question and the lively discussion.